friends, so now I'm back with my next video, and in this video we are going to talk about DNA replication, and we will specifically talk about eukaryotes. So there is the difference between the protein complex and different enzymes involved in the eukaryotes and prokaryotes. So we will specifically talk about eukaryotes. So please make sure to like, share and subscribe my channel. So, what is replication? So, it is the process of making multiple copies or the identical copies of one molecule. So, in case of DNA replication, it's the making identical copies of the DNA molecule, and it occurs based on the Chargaff rules, which says that the cytosines and guanine forms the three hydrogen bonds, and the adenine and thymine forms the two hydrogen bond. So which means that if there is cytosine present in one of the strand, the another step is to make the identical copies or copy of the one of the strand, right? So if we have cytosine on one strand, the another strand will contain guanine, which will pair with the cytosine using the three hydrogen bond. And same for adenine, if it is present in the other strand, it will be thymine, which will form the two hydrogen bond with adenine. And as I have talked about previously in my video, that DNA replicates semi-conservatively, which means that one of the strand act as a template strand to produce another strand, and then both the strand separates, and then one of the strand is from the previous generation and then new strand is formed, which means that it replicates semi-conservatively. And so DNA replication in eukaryotes, it's basically divided into three stages as always. So first is the initiation, in which the initiation complex is formed. Different enzymes are recruited at a particular point to initiate the replication. And then is the elongation process, in which the process from where it started, it elongate in one of the direction. And then the termination, which stops the reaction like the replication, and then the enzymes are removed to produce to act on the another strand. So I will talk about briefly about all these processes, starting with the initiation complex, right? So first step is the initiation of the replication. So it is the first step in which different proteins they come together to form a complex which is known as the pre-replicative complex. So it is the complex before the replication initiates. So this complex is called the pre-replicative complex, and during this process different enzymes or different proteins are involved. So first is the origin recognition complex. So it's a complex of different 11 proteins which binds to the ORIC site, like the origin of initiation site, right? It binds to the complex and then recruit different enzymes and protein. One, now that we have the ORC binding to the site, it recruits the protein CDC6, which is a cyclin division cycle 6, which is a kinase which phosphorylates and which get phosphorylated, and it is important to recruit the another protein which is called the MCM complex and CDT1. So CDT1, it's the chromatin licenses and DNA replication factor 1, and these two both proteins are important for recruiting the Minichromosome maintenance protein complex. So this is a helicase. So the function of the helicase is to separate two strands. So as you know, in case of DNA is a double helix protein, and to initiate the replication, both the strands needs to be separated to act as a template strand for the daughter strand, right? So both the strand get separated and both of them act as a template strand. So this process of separation occurred by the helicase. So helicase is a protein which is involved in the separation of both the strand, and this MCM2 to 7 complex, it's an helicase. So CDC6 and CDT1, they are important for recruiting an helicase to initiate the replication. So as I have talked about the ORC, it binds at the ORI site of the DNA, and then it recruits the CDC6, and then CDC6 recruit the CDT1, which both are the kinases, and they both are important for recruiting the MCM complex, which is an helicase, that is it helps in the separating both the strands. And then MCM finally binds with the chromatin, and all these stages occur in the G1 phase. So as you know the different phases of the cell cycle, so G1 phase is the phase where the preparation for the replication start. So now in until now we have not started any replication, we are just preparing the complex, we are just recruiting everything to initiate the replication. So that's why all these steps occur in the G1 phase. And when everything is done, we move to the next phase of the cell cycle, which is the S phase or the synthesis phase, and it is the phase during which the DNA replication occurs. So here you can see the steps in the initiation. We have a double-stranded RNA, and then the ORC complex binds to this site ARS, and then it recruits CDC6, CDT1, and then these two recruits MCM complex. So here you can see the yellow one are the MCM complex, and this is called the pre-replication complex, and all these steps occur in the G1 phase. When we have this thing ready, the pre-replication complex, we move to the S phase of the cell cycle where the replication starts, right? So then comes the protein kinases, CDKs and DDKs, which phosphorylates this and activate the complex, right? So this complex is still in the inactivation form, they needs to be activated, which is done by the kinases which phosphorylate this complex and activate this complex. So once this complex is activated, you can see the MCS started to separate these strands. So this is one of the strand, right? It forms the bubble kind of structure, which like both of the strand get separated to recruit another enzymes to be activate the initiation. So the MCM complex is very important for the separation of both the strands. So now that the initiation have started, we move to the process which is called the elongation. Now that we have started this initiation, we want to continue this here, like it started in the middle. So now we want to continue in the right and in the left direction, which is done by elongation. So the elongation step is the once the initiation complex is formed, we pass into the S phase, and the complex that becomes activated is called the replisome, and then elongation is initiated. And once the elongation is initiated, it forms the replication fork that we have talked about, right, where the MCM complex is bind by unwinding the DNA. So this fork is called the replication fork because it consists of two strands, and then it forms the two different, like they get separated. And this is the strand from 5 to 3 direction, and then this is the strand with 3 to 5 direction. As I have talked in my previous video that they needs to be in the opposite direction to bind to each other. And the important thing is replication like always occur in the 5 to 3 direction. So here you can see the 5 to 3 strand. So if the replication needs to happen in the 5 to 3 direction, like the daughter strand, this strand, it will form in 5 to 3 direction. It have to start from here. As I have talked about, it needs to be in the opposite direction, right? So if this strand here is a 3 prime, this needs to be 5 prime. So it will start from here 5 prime to 3 prime. And the another way, and the other way is that because this is 3 prime to 5 prime, it have to start here from the 5 prime to 3 prime. So here you can say it's moving continuously, right? But here it cannot move continuously because we have to change every time different strand because it's the opposite direction. It's not the regular direction because it's 3 to 5 prime. So here it occurs in pieces. So this strand is called the lagging strand, and this strand is called the leading strand. So just to make it clear, the leading strand is the strand which is replicating in the same direction as the movement of the replication fork. which means, as I have talked about, the DNA replicates from 5 to 3 direction, right? 5 to 3 direction, and this strand is also replicating in 5 to 3 direction. So that's why this strand is called the leading strand and the opposite strand is called the lagging strand, because this strand is 5 to 3 direction and then this strand is also 5 to 3. So nucleotides in this case are added by the polymerase epsilon, right? And then DNA polymerase, it requires the primer to initiate, because if you have the one strand to initiate the priming, like to add different nucleotide, you need a primer, which is a piece of DNA which consists of some nucleotides which are produced by the primase, and then elongation take place in the 5 to 3 direction, which is normal. And once the replication is done, the primers are removed by the RNase H and the gap is filled by the DNA ligase. So the nucleotides that are added, because they are from the RNA primer, right? But the DNA needs to be totally consist of the double strand DNA, deoxyribose sugar. So they are removed by the RNase H, and then the nucleotides are added by the DNA ligase 1. So we have talked about the leading strand. Now we will talk about the, for the spelling, the lagging strand. So the DNA replication on the lagging strand is discontinuous and it is in the opposite direction to the replication fork, because the replication fork, like the direction of the strand is 3 to 5, but the replication is happening is 5 to 3, so it's in the discontinuous direction. And then in this case, the nucleotides are added by the delta instead of the epsilon. And then the lagging strand used more RNA primers, because as I have talked about, you need to add this stepwise for loading the nucleotides. And then DNA polymerase will initiate small fragments because it's needing multiple primers. So these fragments are called the Okazaki fragments, which are joined by the which are added to the 3 prime, and then they are joined by the ligases. So here you can see that elongation complex, we have the origination complex binding, right? We have the bubble forming, which is called the replication fork, and then we need the primers to be added to this, right, which are provided by the primase, which are mostly RNA primers. And then in the leading strand, this is occurred by the epsilon, and then in the lagging strand, it's done by the delta. So here you can see from the 5 to 3 direction, if it's moving in also in the same direction, then this is called the leading strand. And if it's going in the opposite direction, like here from 5 to 3, right, this is the leading strand. But if we are going from 3 to 5, then it's opposite, and then we need to be then it needs to be the Okazaki fragments, which are then joined by the ligases, right? So this is the elongation complex. So now once we have the elongation done, like when all this strand is covered, we move to the next phase, which is the termination of the DNA replication. So the termination occur by the telomeres regions and telomerases. So what are telomers and what is telomerases? So telomeres extend the 3 end of the parental chromosome. So as the replication moves from 5 to 3, so there will be some point when the replication, like the leading strand stops, like we don't have anything, right? But the replication continues and we need to stop that. It extends the 3 end of the parental chromosome beyond the 5 strand, right? It like occur on the parental chromosome, and then it recruits the telomerase, which is a polymerase, consists of a multiple protein subunit and the RNA component. So RNA component of this anneals to the single strand of the template DNA, and then contains the telomeric sequence. And then telomerase, it's a reverse transcriptase, which called the TERT, or which synthesize DNA until the end of the template telomerase RNA and disengages. So it's just the text, but I will explain how it works, right? So you have this strand, the template strand and the daughter strand, right? So in this end of the 3 prime, the telomers bind, right? So this is the sequence of the telomers, which binds, which recruits the telomerase, right? So telomerase contains a RNA and protein subunit. This RNA, it binds to this three strand. It is complementary to these three strands of the DNA, right? And then it starts adding nucleotides to this end, right? So first step is the binding of the telomerase to the single strand of the eukaryotic DNA, and then it occurs the polymerization by adding different nucleotides which are complementary to this telomeric RNA, right? And then telomerase, once it gets recruited 6 nucleotide, it moves to the right and begin to make another repeat. So these repeats are important to determine the, like, to prevent the actual DNA from degradation and everything, because in every replication there is some loss of the part of the DNA from the ends, or there is some degradation. So we make enough repeat so that the major portion of the DNA which codes for protein does not get degraded. So it just polymerize like it, and after every 6 nucleotide it moves to the right and begin to make another repeat. And then once it completes its task, it disengages from this 3 prime end, right? And then the complementary strand is added by the primase, which can be RNA primer, and then this is joined by the DNA ligase. So this is the step where we add another repeats, and once this is finished, we have the completely DNA strand, which are the ends are the telomeric DNA or telomere sequence, which prevents them from degradation. So this is in short the initiation, elongation and termination. I hope you understand it, and if you have any doubts, please let me know in the comment section, and please make sure to like, share and subscribe my channel. Thank you.